So good morning all of you and welcome back to Excelsure Academy. So today we are going to study about the after cell lysis, what are the steps required to carry out protein purification. Okay. So already we have talked about cell lysis. So where we mentioned about the mechanical and non-mechanical. So if you have seen the video, you must be thorough with that. So where non-mechanical means it is going to have both physical, chemical and enzymatic method. Now, among all these three, enzymatic methods are important because you have to remember the names of the enzymes required whenever you're dealing with bacterial cell or animal cells or plant cells. So bacterial, it is lysozyme, animal, it is trypsin, and plant, it is cellulases. And sometimes few people also use hemicellulases. Okay, so this is important to remember. Now we have about the crude extract preparation. So once you lyse the cell, okay, the membrane, the cell wall has been disrupted. What is the next step? You need to centrifuge it. So what you're going to do, take your sample into the falcons. So this kind of falcons you will get. It has got the blue cap, plastic material, and then use it for centrifugation. Okay. Now remember, once the bacterial cell or any cell has been ruptured, everything you need to carry it out at low temperature because your proteins are fragile and they might get disrupted. So when you centrifuge also, it actually generates heat, right? Because a force is there. At very high speed you are rotating. So what happens? Heat is generated, right? And because of this, you need to keep your sample, everything at low temperature. Now, next. So, fractionation and ammonium sulfate precipitation and the pH one. So, this is what we are going to study in detail today. Okay. Yes. So, ammonium sulfate precipitation. So, most commonly used methods, it is for your large and laboratory scale protein purification and fractionation. Okay, so it is used to separate proteins by altering their solubility in the presence of high salt concentration. So what exactly we are doing over here? We are going to add ammonium sulfate to the protein solution. Okay, so the extract what we have got, we are going to add into that. Now, before that, ammonium sulfate, we should know that it is an inorganic salt and it has got high solubility because of which dissociates into the ammonium as well as sulfate in aqueous solution. So what is happening? The moment you add ammonium sulfate in water, it dissociates into ammonium ion as well as sulfate ions. Okay. Now it is useful as a precipitant because it is highly soluble. It stabilizes protein structure. This is important. It has got relatively low density, readily available, and is inexpensive. So this is actually a very cheap way of doing protein precipitation. Now, what is the mechanism behind that? Now, the proteins are stabilized by solvation. What do you mean by this? It means that if you have a protein like this, so every time it has been surrounded by water molecule. Okay. So these are your hydration shell. It is your hydration shell where this is your protein. Okay. So proteins are stabilized by solvation. It is nothing but your water solvation hydration shell. It also helps in misfolding proteins to fold back into its native form. So now what we are doing is, as the protein solubility increases with increasing salt concentration, it is called salting in. So what you are doing exactly over here? You keep on adding ammonium sulfate to your protein solution. So what is happening? The protein solubility is increasing. Now how this is happening? So you have got your protein over here, another protein over here, another protein over here. Okay. Now each protein has got its water hydration. Now what happens is this ammonium sulfate will remove these water hydrations and will bring protein molecules together. So something like this it will look. So what is happening? All your proteins are coming together. Okay. So this is how the salting out will happen. See over here. As the salt concentration is further increased, the solubility of the protein begins to decrease. It means what? It will no longer be present like this. Individual proteins in the solution. So at a sufficiently high ionic strength, when you have added a high amount of ammonium sulfate, the protein will precipitate out, and this is called salting out. So what exactly the principle or the mechanism we are using here is the salting out process. Okay. So now what will happen? More and more of protein will come. More and more of protein will come over here. Okay. And then what happens? All these will come together and it will precipitate out. Now, what will happen? Next, what you have to do is centrifuge. Now, once you centrifuge, what will happen? You will have the supernated and the pellet. Now, this pellet is nothing but your protein precipitate. Okay. Now, when the ammonium and sulfate ions are within the aqueous solution, they are attracted to opposite charges as evident on the compound that is being purified. So what does it mean? See, even your proteins are going to have some positive and negative charges. So correspondingly, if your protein is having negative charge, ammonium will go in by positive charge wherever sulfate is there. So this is how it surrounds your protein. Now the attraction of opposite charges prevents water molecules from the interacting with the compound being purified. So it leads to salting out. So what does it mean? So whenever protein is there, water molecules are there. So when these negative and positive charge of ammonia go and interact, it removes these water molecules. Okay. So your water hydration shell is removed. Okay, so it is removed by the ammonium sulfate. Now, with the mechanism of salting out, there is an omission of the salt from the layer of water, which is closely associated with the surface of the protein known as hydration layer. So this is what I was talking about. Now, this plays an important role in solubility and suitable natural conformation. So there are three main protein-water interactions. So what are those? Ion hydration between the charge side chains. So you have a protein. Each amino acid has got their side groups, right? So they are interacted with the water molecules. Hydrogen bonding between polar groups and the water. So you know that many amino acids like serine or threonine, they have got OH group. So they form hydrogen bonds with the water molecules which are present in the solution. Then hydrophobic hydration. So what is hydrophobic hydration? So many proteins are there having the hydrophobic amino acids. So they will come together. So they will cluster. So all these three are responsible for maintaining the native conformation of protein. Okay. So what do you mean by native conformation? The original folded structure of the protein. Please stop me if something is not clear. Okay. Now, once the salt is added to the mixture, there is an increase in the surface tension of water. So, what will happen if there is an increase in the surface tension of water? The hydrophobic interactions between the water and protein of interest will increase. So, what does it mean? When your proteins are there, the hydration shell is getting disturbed and it will result in clustering of proteins. Okay, like this. The protein of interest then reduces its surface area, which diminishes its contact with the solvent. And what will happen? All the proteins will start coming together, increasing the hydrophobic interactions. Now, this is also shown by the folding and self-association, which ultimately leads to precipitation. So, even if your proteins are suppose unfolded during your crude extract preparation, if you add ammonium sulfate, what happens? It actually tends your protein to go into correct folding conformation. So it helps in protein folding also. The folding and self-association of the protein pushes out free water. So whatever the water molecules are present surrounding them, it will be pushed, leading to increase in entropy and making this process energetically favorable. Now this is important because the questions can be asked is that addition of ammonium sulfate is spontaneous or non-spontaneous. Okay. So having ammonium sulfate precipitation reaction. So the answer should be that it is ammonium sulfate precipitation of protein is actually a spontaneous process. Okay. Please remember this point. It is important. So I repeat, the folding and self-association of the protein pushes out free water, leading to an increase in entropy and making this process energetically favorable. Okay. Yes. Now the procedure how you carry out. So the percentage of ammonium sulfate is increased stepwise and the precipitated protein is recovered at each stage. So remember in the previous class I have mentioned, so you have got a crude extract protein. Okay, so crude extract is there, you have centrifuge. Now suppose this is 100 ml. So I have mentioned that you have to start with 10% and go till 90%. So what you're going to do, the amount of sodium sulfate, the amount of ammonium sulfate that should be added to the solution can be determined from published nomograms or by using online calculator. So many online softwares are available in which you just have to add the values of temperature, initial volume of your sample, and initial concentration of the ammonium sulfate, which is going to be 0% since we are just going to start, and then required percentage, so 10%. So that online software will tell you how much gram of ammonium sulfate you need to add. Okay. Now, let us say that we are going to start with 10 gram in 100 ml of protein extract. Now, what you will do? You just have the beaker. You have got your sample in it. Okay. So, sample is over here. Then, this is kept on the stirrer. I hope everyone knows what is stirrer. It's a machine in which you rotate like this and your sample moves like this. Okay, so it just helps in the mixing. So slowly you keep on adding your ammonium sulfate. Now everything has to be done at low temperature. Do not forget low temperature. So how much low temperature are we talking about? 20 degrees Celsius. So either you can keep your AC on in the lab or you can go to cold room or you can go to cold room. Now once this mixing is done, keep it for 1 hour undisturbed. Okay. Now please remember these steps are important. Now once you keep it for 1 hour, what will happen? All the precipitate which was present on the top, right? All the precipitate which is there, which is getting mixed. What happens? Because of the precipitation out, the proteins will then settle down. Okay. So then what you have to do is take the beaker, put into the falcons, like falcons I showed you, which is used in the centrifuge. So just measure it, make it a balance, put into the centrifuge and rotate. So what is the speed you're going to use in the centrifuge? It is around 10,000 RPM, roughly around 20 minutes. Okay. So these are the standard steps which we use in the labs. Okay. So once this is done, then what you have to do is take out the supernatant and the pellet. So Ramya, which one is of our interest, supernatant or pellet? What should I save it for future use? Is it the supernatant or the pellet? Yes, Ramya?
Pellet, ma'am.
Yes, very good. So I'm asking in between so that I am sure that you people are listening. So very good. It is the pellet. So your pellet is nothing but your protein precipitate. Okay. Yes. Now what you have to do is take this protein pellet. Take this protein pellet. Now dissolve in your required buffer. Okay. So now when you dissolve it in required buffer, what will happen? You have to go for another step which is called dialysis. Okay. So now let's talk about the dialysis. So this is the same thing which I have just mentioned. So in the next stage of purification, all the salts needs to be removed from the protein, for which we are going to do the dialysis. Now what is dialysis? It is the process of separating molecules in solution by the difference in the rates of diffusion. So this we all know what is dialysis through a semi-permeable membrane. So in research lab, we use dialysis tubing. And I have mentioned also what is dialysis tubing. So actually you get a roll like this, like how you have a tissue roll, right? So how much you need depends on your protein solution. So if you have got around 2-3 ml of sample, okay, 2-3 ml of your protein precipitate sample, you just need around 5 to 6 centimeter tubing. So I'm going to make a cut like this. So here it is open and here it is open. So then with the help of the clamp, I'll just seal one side. Okay. And from here, the another side, I'm going to add the protein precipitate, which is dissolved in the buffer. So what will happen? This is all filled with the protein sample. Again, I'm going to seal it. Now this tubing is nothing but it's a semi-permeable membrane. So we know that whenever there is a concentration gradient formed, the molecules will move from high concentration to low concentration. Okay. Now here it is protein molecules we are talking about. So the protein molecules are going to be larger in size. So they cannot move from inside of the dialysis bag to the outside. So what is going to move? It is the water of the buffer. So buffer which is present inside this is less and outside in the beaker it is more. So the buffer is at high concentration. So buffer is going to move from outside to in. Okay. Till equilibrium is reached. Now, your dialysis membrane are made up of cellulose or synthetic polymer like cellulose acetate or nitrocellulose. Okay. So this is also important to remember, like of what kind of material your dialysis membrane is made up of. Okay. So this is the last. So what we have done, so already the figure has shown that, figure shows you that already clamp is there, right? So I have made the clamp so that the protein sample does not leak out. Okay. Now, so this is your bag and inside you have got the concentration solution. This is your buffer and inside buffer also has to be same. Now, what happens if the pH is different? What will happen if the pH is different? Any idea? Ranjan, what will happen if the pH is different inside this and over here? Suppose this is 8 and this is 6 pH. What will happen? So these kinds of questions are asked during the interviews. Yes, tell me what will happen if the pH is different? Anyone? Pallavi, try. Yes. So
The
till now,
solution may
huh?
the solution may come out, ma'am.
No. See, it has been sealed over here properly in both the sides. So what will happen if the pH is 6 and 8? There is no leakage of the solution. Nothing will happen to the leakage. Yes. Anyone else? Ramya? Think about it. Why every time I was telling that the pH needs to be same?
To maintain the equilibrium.
No. No. Anyone else? Last try. What is the important thing we are doing in dialysis? Why we are studying all these things? For what? Who is the hero over here? Okay, in another words, what I should save? Ranjan, yes.
Protein.
Exactly, it is the protein, right? So if you change the pH of the solution, what happens? The pH will affect the protein solubility as well as the structure. Remember? So if there is a change in the pH all of a sudden, see, dialysis means what? Here the pH was less, and all of a sudden you are just dipping into the beaker containing different pH solution. So what will happen? The protein will precipitate, and this precipitation is not good. Ammonium sulfate precipitation is good, okay? But if suppose you precipitate like this because of difference in the pH, what happens is it loses its structure and protein loses its activity, okay? So protein will get denatured if there is a change in the pH. Okay, similarly with the temperature also. So if you were if you were doing the experiments at if you are doing at the low temperature 20 degrees Celsius, all of a sudden you go to 40 degrees Celsius, again the protein will get disrupted. Okay, so every time whatever we are doing only for the sake of the protein stability, because at the end of the day of all the experiments I need my protein to be functional. There is no point if my protein gets disrupted. Okay, now it is clear. Yes, so always you need to have optimum temperature and pH. Okay, yes. So with this, one second, yeah, so with this I complete the topic over here, and thank you for listening.